Good day, class. I welcome you to study session 8 of biochemistry, which will discuss gluconeogenesis, glycogenesis, and glycogenolysis. Please sit back to enjoy this lecture. In study session 7, we have been introduced to glycolysis. Another interesting topic, gluconeogenesis and glycogenesis, are about to be discussed. At the end of this study session, you should be able to explain the basic concept of gluconeogenesis. You should be able to discuss regulation of gluconeogenesis, explain glycogenesis and glycogenolysis. First, let's look at gluconeogenesis. Gluconeogenesis is a metabolic pathway that results in the generation of glucose from non-carbohydrate carbon substrates such as pyruvate, lactate, glycerol, glucogenic amino acids, and fatty acids. Gluconeogenesis is often associated with ketosis. It is one of the two main mechanisms humans and many other animals used to keep their blood glucose levels from dropping too low. The other means of maintaining blood glucose levels is through degradation of glycogen, that is glycogenolysis. In vertebrates, gluconeogenesis takes place mainly in the liver and to a lesser extent in the cortex of the kidneys. In ruminants, this tends to be a continuous process. In many other animals, the process occurs during periods of fasting, starvation, low carbohydrate diet, or intense exercises. Gluconeogenesis is also a target of therapy for type 2 diabetes, such as metformin, which inhibits glucose formation and stimulates glucose uptake by the cells. What are the precursors in gluconeogenesis? In humans, the main gluconeogenic precursors are lactate, glycerol, alanine, and glutamine. Lactate is transported back to the liver where it is converted into pyruvate by the Cori cycle using the enzyme lactate dehydrogenase. Transamination or deamination of amino acid facilitates entering of their carbon skeleton into the cycle directly or indirectly via the citric acid cycle. Pyruvate, which is the first designated substrate of gluconeogenic pathway, can be used to generate glucose. In mammals, gluconeogenesis is restricted to the liver, the kidney, and possibly the intestine. However, these organs use somewhat different gluconeogenic precursors. The liver uses primarily lactate, alanine, and glycerol, while the kidney uses lactate, glutamine, and glycerol. Regulation of gluconeogenesis. The rate of gluconeogenesis is ultimately controlled by the action of a key enzyme, which is fructose 1,6-biphosphatase, which is also regulated through signal transduction by cyclic AMP and its phosphorylation. Most factors that regulate the activity of gluconeogenesis pathway do so by inhibiting the activity or expression of the key enzymes. However, both acetyl-CoA and citrate activate gluconeogenesis enzymes. While most steps in gluconeogenesis are the reverse of those found in glycolysis, three regulated and strongly hexagonic reactions are replaced with more kinetically favorable reactions. Hexokinase, phosphofructokinase, and pyruvate kinase enzymes of glycolysis are replaced with glucose 6-phosphatase, fructose 1,6-biphosphate, and PEP carboxylase. This system of reciprocal control allows glycolysis and gluconeogenesis to inhibit each other and prevent the formation of a futile cycle. Recent studies have shown that the absence of hepatic glucose production has no major effect on the control of fasting plasma glucose concentration. Compensatory induction of gluconeogenesis occurs in kidneys and intestine, driven by glucagon, glucocorticoids, and acidosis. Let's go into glycogenesis. Here, glycogenesis occurs when glucose concentrations are high, and it involves 4 steps. 1. Glucose is phosphorylated to glucose 6-phosphate. Then this glucose 6-phosphate is isomerized to glucose 1-phosphate. UDP is added to glucose 1-phosphate to form glucose UDP, and glucose UDP is added to the growing glycogen chain, as you can see on your screen, to showing the process of the synthesis of glycogen. Glycogenolysis. Glycogenolysis is the breakdown of glycogen to glucose 1-phosphate and glycogen. The overall reaction of the breakdown of glycogen to glucose 1-phosphate is represented by the chemical reaction on the screen. Also, glycogen phosphorylase cleaves the bond linking a terminal glucose residue to glycogen. by substituting a phosphoric group for the alpha 1,4 linkage. Glucose 1 phosphatase is converted to glucose 6 phosphatase by the enzyme phosphoglucomutase. Glucose residues are phosphorylized from branches of glycogen until 4 residues before a glucose that is branched with alpha 1,6 linkage. Glycogen debranching enzyme then transfers 3 of the remaining 4 glucose units to the end of another glycogen branch, which exposes the alpha 1,6 branching point, which is hydrolyzed by alpha 1,6 glucosidase, removing the final glucose residue of the branching as a molecule of glucose and eliminating the branch. Glycogenolysis takes place in the cells of the muscle and liver tissues in response to hormonal and neural signals. In particular, glycogenolysis plays an important role in the fight or flight response and regulation of glucose levels in the blood. What is the clinical significance of this? The parenteral administration of glucagon, which is a common human medical intervention in diabetic emergencies when sugar cannot be given orally, and it can also be administered intramuscularly. Thanks for listening. Have a good day.